Okay, let's unpack this. Have you ever wondered what's truly going on behind the scenes when some microscopic invader makes you sick?
Mm.
It's not just, a germ getting into your body. It's this fascinating, really intricate battle of strategies, often playing out on a scale we can't even see.
Right.
Today, we're taking a deep dive into the incredible microbial mechanisms of pathogenicity.
And what's really fascinating here, I think, is that these microorganisms, they aren't consciously trying to cause disease in us. Not really.
Right, they're just trying to live.
Exactly. From their perspective, they're simply looking for nutrients, defending themselves. But sometimes, just their presence, or even tiny parts of them, can trigger some pretty profound symptoms in a host, in us.
Okay.
So we're going to explore how they manage this intricate dance of overcoming our defenses and, well, causing damage. And importantly, what it all means for you and your health.
Our mission today, then, is to pull out the most important insights from, a comprehensive look at how microbes cause disease, starting from when they first get in, right through to how they eventually get out.
And tree to exit.
You're going to get, hopefully, a clear understanding of the major concepts, these clever microbial processes, the structures they use, maybe some surprising lab techniques, key diseases, and how all this actually applies in the real world.
Yeah, there are some real-world connections here.
Get ready for some serious aha moments.
Really?
So, to start us off, let's maybe lay some groundwork. We hear the term pathogen all the time, but what precisely makes a microbe, pathogenic?
Right. So pathogenicity, basically, it's just a microorganism's ability to cause disease. Its potential.
Yeah.
And it does that by overcoming a host's defenses.
The potential. Okay.
Think of it as the potential to harm. Now, the degree or the extent of that ability, that's what we call virulence.
Ah, virulence. Okay, so ability versus degree.
Exactly. And if we connect this to the bigger picture, it's a really dynamic interplay. Think about the cholera pathogen, Vibrio cholerae.
Yeah, nasty.
Right. It rapidly causes severe diarrhea, which is obviously life-threatening for the host. But from the microbe's point of view, this awful symptom, it's also an incredibly effective way to transmit itself.
How so?
By contaminating the environment, the water supply, it helps it spread to new hosts. It's a remarkable, though, unfortunate example of coevolution. The pathogen's survival relies on how it impacts its host.
Wow, that's ruthless, but kind of brilliant, evolutionarily speaking. So it's not just about surviving inside, it's using the symptoms to spread.
Precisely.
Okay, so once a microbe has this ability, this pathogenicity, and maybe a certain degree of virulence, how does it actually get inside us? Where are the main doors, so to speak?
Yeah, the main entry points, we call them portals of entry. The most common ones are our mucous membranes, our skin, and then there's something called the parenteral route. Yeah, it means direct deposition beneath the skin or membranes, bypassing the surface barriers.
Okay, let's break those down. Mucous membranes first.
Right. These soft, moist linings are prime entry points. Think respiratory tract, probably the easiest and most common.
And breathing stuff in.
Exactly. Microbes get inhaled in tiny moisture droplets or dust. That leads to familiar things like the common cold, pneumonia, flu, measles.
Makes sense. What else?
Then there's the gastrointestinal tract, eating or drinking contaminated stuff. Now, most microbes thankfully get destroyed by stomach acid or enzymes, but those that survive can cause serious illnesses like typhoid fever or cholera, and they often get eliminated via feces, which, again, helps them spread.
Right, like the cholera example.
Then the genitourinary tract is the gateway for STIs, sexually transmitted infections like HIV, syphilis, gonorrhea, and even the conjunctiva, the membrane lining your eyelids and eyeball, can be a portal. Think pink eye?
Conjunctivitis, yeah. Okay, what about skin? Isn't that supposed to be a barrier?
It is. It's a pretty formidable barrier, actually. But some microbes find tiny openings, like hair follicles or sweat gland ducts.
Clever.
And some are even tougher. Hookworm larvae, for instance, can actually bore right through intact skin. And certain fungi just grow directly on the keratin in our skin, athlete's foot, that kind of thing.
Right. And that last one? Parenteral.
Parenteral route. That's when these barriers are bypassed entirely. Direct entry into tissues.
How does it happen?
Punctures, injections, bites, insect bites, animal bites, cuts, wounds, even surgery.
Ah, okay.
Diseases like tetanus, gangrene, and viruses like HIV and hepatitis can all get in this way.
So it sounds like it's not just where they get in, but also how many of them get in, and maybe if it's their preferred route, does that preference really matter much?
Oh, absolutely it does.
Right.
Many pathogens have a very specific preferred portal of entry. Well, take Salmonella typhi, the bacteria causing typhoid fever. It causes a severe illness when you swallow it, but if you just rub it on your skin, probably little to no reaction. Or Streptococcus pneumonia causes pneumonia when you inhale it, but it doesn't usually cause problems if you swallow it.
So the route is critical.
It is, and this preference highlights a crucial part of their virulence. And we actually quantify this. We use two key terms, ID₅₀ and LD₅₀.
ID₅₀, LD₅₀. Okay, what are those?
ID₅₀ is the infectious dose for 50% of a sample population. Basically, how many microbes does it take to make half the exposed individuals sick?
Okay, and LD₅₀.
That's the lethal dose for 50%. How much of a substance, like a toxin, or how many microbes does it take to kill half the test population?
It gives you a measure of potency or danger.
Exactly. For example, Bacillus anthracis, the anthrax bacterium. The ID₅₀ for getting it through your skin, cutaneous anthrax, is just 10 to 50 endospores. Tiny number.
Very low.
But for gastrointestinal anthrax, swallowing it, you need maybe 250,000 to 1,000,000 endospores. A huge difference.
Clearly, skin exposure is much easier, much more dangerous in that sense.
Right. And when we talk about toxins, LD₅₀ shows their sheer power. Botulinum toxin, the Botox toxin, has an LD₅₀ of just 0.03 nanograms per kilogram in mice.
Nanograms. That's minuscule.
Astonishingly potent. Far, far more potent than something like Shiga toxin or staphylococcal enterotoxin.
Wow. Okay, so they've found their preferred way in. Maybe enough of them got past our first lines of defense. What's the very next step? They can't just float around, can they? They need to stick.
That's exactly right. Adherence is absolutely necessary. They need to attach.
How do they do that? Velcro?
Sort of, on a molecular level. They use specialized surface molecules called adhesins or ligands.
Adhesins?
Yeah, usually proteins or maybe lipoproteins. And these bind specifically to complementary receptor sites on our host cells, often sugars, actually.
Like a key in a lock.
Precisely. A molecular key fitting a very specific lock. These adhesins can be located on the microbe's glycocalyx, that's its sticky outer coat, or on little hair-like things called pili or fimbriae, or even on their flagella, the tails they use to swim.
Can you give an example?
Sure. Streptococcus mutans. It's a key player in tooth decay.
Oh, cavities.
Right. It uses its glycocalyx, which is made of a sticky sugar called dextrin, to attach firmly to your teeth. Then other bacteria, like actinomyces, can stick to S. mutans' glycocalyx, and this builds up into
Plaque.
dental plaque, exactly, which is a perfect example of a biofilm.
Biofilm, I've heard that term. What exactly is it?
Biofilms are these incredibly organized microbial communities. They cling to surfaces, they share nutrients, and maybe most importantly, they're remarkably resistant to things like disinfectants and even antibiotics.
Really form protective communities.
Yes. Dental plaque is a common one, but biofilms are a huge problem in medicine too. They colonize critical things like catheters, stents, artificial heart valves.
That sounds serious.
It is. It's estimated that biofilms are involved in something like 65% of all human bacterial infections. They're a major challenge.
That's fascinating how they cooperate and build these fortresses. Okay, so they're stuck, maybe even building communities. Now comes the really hard part, right? Dealing with our immune system.
The defenders.
What are some of the first tricks they use to evade our body's best defenses?
Well, a major evasion strategy involves capsules. Many bacteria form this glycocalyx layer that creates a slick protective capsule around their cell walls.
A slimy coat?
Sort of, yeah. And this capsule is very effective at resisting phagocytosis.
Phagocytosis, that's when our immune cells eat the invaders, right?
Exactly. Cells like macrophages try to engulf and destroy microbes, but the capsule's chemical nature often prevents these phagocytes from getting a good grip from adhering to the microbe.
So they slip away.
Pretty much. For instance, the really dangerous virulent strains of Streptococcus pneumonia, they have capsules. The harmless strains usually don't.
Ah, key virulence factor then.
Definitely. Other well-known bacteria like Klebsiella pneumonia, which causes a type of pneumonia, and Bacillus anthracis, anthrax again, also use capsules as a primary shield.
Okay, so the capsule is one major shield. Are there other parts of the actual bacterial cell wall that act as defenses or even weapons?
Absolutely. Certain components built right into the cell wall contribute significantly to their ability to cause disease, their virulence.
Like what?
For example, Streptococcus pyogenes, the strep throat bug, has this protein called M protein on its surface and on its fimbriae.
M protein.
Yeah, it's tough, resistant to heat and acid. And it does two things. helps the bacterium attach and helps it resist being eaten by phagocytes. Our immunity to strep actually depends on making antibodies specifically against this M protein.
So we have to target that specific protein.
Right. Then you have Neisseria gonorrhea. It uses its fimbriae, those hair-like things, and another outer membrane protein called OPA to attach to and enter our host cells. They basically trick the cell into taking them in.
Invite themselves inside.
And maybe one of the most interesting examples is the waxy lipid mycolic acid found in the cell wall of Mycobacterium tuberculosis.
The TB bacterium.
Yes, that waxy coat actually allows it to resist digestion after it's been engulfed by our immune cells, by phagocytes.
So it survives inside the very cell meant to kill it.
Exactly. It can even multiply inside them. It's like having a secret hideout within our own defenses.
It really sounds like they've got their armor sorted. But some microbes also seem to carry, well, an entire biochemical toolkit, don't they? These enzymes.
Yeah.
It's fascinating how they can actively manipulate our body's own systems.
Indeed. They produce these powerful extracellular enzymes, we call them exoenzymes, that really boost their virulence. They're secreted outside the cell.
What kinds of things do they do?
Well, take coagulases. Some Staphylococcus species make these. They actually coagulate fibrinogen, a protein in our blood, essentially forming a fibrin clot around the bacteria.
Like building a wall.
Exactly. A protective clot that can shield them from phagocytosis, isolate them from our immune defenses.
Clever. What else?
Then, conversely, you have kinases, like streptokinase, which is made by Streptococcus pyogenes. These enzymes do the exact opposite. They break down fibrin clots.
Why would they do that?
To escape. It allows the infection to spread rapidly from a localized area, breaking out of any clot that might contain it. In fact, streptokinase is so effective, we actually use it as a medicine.
Really?
Yeah, therapeutically, to dissolve dangerous blood clots in heart attack patients.
Wow, using the microbe's weapon against our own clots.
Then there's hyaluronidase, secreted by streptococci and also some Clostridium species, the ones that cause gas gangrene.
Yes, gangrene, nasty stuff.
Very. Hyaluronidase breaks down hyaluronic acid. You can think of hyaluronic acid as the glue holding our connective tissues together.
So it dissolves the body's cement.
In a way, yes. By breaking it down, the microbe can spread much more rapidly through tissues. It contributes to that awful tissue damage and blackening you see in gangrene.
Okay. What else is in the toolkit?
Collagenase, also produced by several Clostridium species. It breaks down collagen, which is the main structural protein of connective tissue, like tendons and cartilage. Again, this helps the gas gangrene spread.
So they're literally dissolving the body's structure to move through it.
Right. And one more, some pathogens, like Neisseria gonorrhea, again, produce IgA proteases.
Protease breaks down protein.
Exactly. Specifically, they destroy IgA antibodies. IgA is a crucial antibody type found on our mucosal surfaces lining the respiratory tract, the gut. It's a key first line of defense there.
So they're disabling the border guards.
Precisely. Dismantling the watchtowers at the border, you could say.
It really is like a microscopic arms race. They're not just defending, they're actively attacking and manipulating our systems. And just when our body, mounts an immune response, figures out what they look like, some microbes pull an incredibly sneaky trick. Tell us about antigenic variation.
Yes, antigenic variation. This is where it gets really interesting and, frankly, quite frustrating for our immune system.
How does it work?
Okay, so our adaptive immune system, the specific part, produces antibodies. These antibodies target very specific molecules, antigens, on the surface of pathogens, like recognizing a face.
Right, unique identifiers.
Exactly. But some pathogens have evolved the ability to change those surface antigens. They can alter their appearance.
Like changing masks.
Perfect analogy. By the time our body produces a whole army of antibodies against one set of antigens, one mask, the pathogen has already switched to a different one. Those antibodies are now useless.
So the immune system is always one step behind.
Often, yes. The classic example is the influenza virus, the flu virus.
That's why we need a new flu shot every year.
That's precisely why. The virus constantly changes its surface antigens. Other masters of this include Neisseria gonorrhea again.
One keeps coming up.
It's very adept. And also Trypanosoma bruchgambiense. That's the protozoan parasite that causes African sleeping sickness.
Sleeping sickness.
Yes. And Trypanosoma is incredible. It's thought to be capable of making up to 1,000 different antigens, a huge wardrobe of masks.
1,000?
One guy. Exactly. It explains why those infections can persist for decades, constantly staying ahead of the immune response.
So they can shield themselves with capsules, use enzymes to break through tissues, and even change their identity with antigenic variation.
Yeah.
But some go even further, right?
Yeah.
Actually getting inside our cells. How do they manage that breach?
Right, invading the host cell itself. Many microbes cleverly trigger the host cell's own internal machinery to let them in.
They use our own systems against us.
They do. They produce surface proteins called invasins. These invasins interact with the host cell's cytoskeleton, specifically the actin filaments.
The cell's internal scaffolding.
Exactly. They cause the actin filaments to rearrange. For instance, Salmonella typhimurium makes the host cell membrane kind of ruffle up.
Ruffle?
Yeah, it creates these waves or protrusions on the cell surface. The bacterium then essentially sinks into this ruffle and gets engulfed by the cell.
Wow. And once they're inside.
Once inside, some, like Shigella and Listeria, take it a step further. They actively hijack the host's actin again to propel themselves. They use the actin to build little tails, allowing them to rocket through the cell's cytoplasm. They can even use this to punch directly from one host cell into an adjacent one without ever having to go outside again.
Staying hidden the whole time.
Pretty much. And what's more, some bacteria have evolved really sophisticated ways to survive inside phagocytes, those immune cells that are supposed to eat and destroy them.
Like the TB example.
Yes. But there are others too. Coxiella burnetii, which causes Q fever, is amazing. It actually needs the harsh, acidic environment inside a phagolysosome, the compartment where digestion normally happens to replicate. It thrives in there.
That's counterintuitive.
It is. Others, like Listeria and Shigella again, they just escape the phagosome, the initial little bubble they're trapped in, before it can fuse with the destructive lysosome.
They break out early.
And Mycobacterium tuberculosis in HIV, they can prevent that fusion process altogether. They just stop the phagosome from merging with the lysosome, allowing them to multiply safely inside the phagocyte.
Incredible survival tactics.
And remember biofilms. We talked about the sticky community.
Yeah, the protective slime cities.
Well, bacteria within a biofilm are much more resistant to being eaten by phagocytes, too. The sticky goo, the EPS, can physically shield their antigens, or sometimes it even contains substances that kill the phagocytes directly. Pseudomonas aeruginosa is known to do this.
So many layers of defense and evasion. Okay. The microbes have successfully entered, they've adhered, they've dodged or disabled our defenses, maybe even set up shop inside our own cells.
Right.
Now comes the damage. How do they actually hurt us?
Right, the actual harm. Pathogens damage host cells primarily in four ways. First, by using the host's nutrients. Second, by causing direct damage near the site of invasion. Third, by producing toxins. And fourth, by inducing hypersensitivity reactions, basically triggering an excessive immune response. We'll focus mainly on the first three today.
Okay, let's start with using our nutrients. How does that cause damage?
Well, one key nutrient is iron. Iron is absolutely essential for the growth of most pathogenic bacteria.
We need iron too, though.
We do, but our bodies are smart. We keep the levels of free iron incredibly low. Most of it is tightly bound to proteins like transferrin in the blood or hemoglobin in red blood cells.
So the bacteria can't easily get it.
Not easily. So to steal this vital nutrient, some pathogens secrete these specialized molecules called cytophores.
Cytophores, iron carriers.
Exactly. These proteins bind iron even more tightly than our own proteins do. Then the whole iron cytophore complex gets taken up by the bacteria. They basically snatch it away.
Resource theft on a molecular level.
Pretty much. And this highlights an important clinical consideration. Think about a liver transplant patient who's also anemic. Why might their doctor stop giving them iron supplements?
Because the extra iron could feed any potential opportunistic infection, give the bacteria the fuel they need.
Precisely. It could inadvertently fuel pathogen growth. Okay, second mechanism. Direct damage.
This sounds more straightforward.
It is. Once pathogens get inside a host cell or even just attach closely, they start using the cell's nutrients, they multiply, and they produce waste products.
Crowding the place out and making a mess.
Basically, yeah. This often disrupts the host cell's function and can eventually cause it to rupture and die. Viruses obviously do this all the time as part of their replication cycle. Intracellular bacteria or protozoa commonly do it too. They multiply, burst out, and then spread to infect more cells.
And even if they don't get inside.
Some bacteria, like certain strains of E. coli or Shigella, can induce host cells, say in the intestinal lining, to engulf them. Then they pass right through the cell and exit on the other side, damaging the cells as they transit through the tissue layer.
Like punching through a wall. Okay, but the most maybe notorious way microbes cause damage seems to be through toxins.
Ah, yes, toxins, definitely a major weapon.
What exactly are toxins? And you mentioned before, there's a difference between an intoxication and an infection.
Right. So toxins are simply poisonous substances produced by microorganisms. The ability to produce them is called toxigenicity. And if toxins get into the bloodstream, that's called toxemia.
Toxemia, toxins in the blood. Got it.
Now, an intoxication is an illness caused only by the presence of a preformed toxin. It's not caused by the microbe actually growing and multiplying in your body.
Example.
Botulism is the classic one. Usually you get sick from ingesting the toxin that the bacteria already produced in contaminated food, not from the bacteria themselves setting up an infection. Staphylococcal food poisoning is another common intoxication.
Okay, so toxin first, illness follows, no infection needed. What about the toxins produced during an infection?
Those fall into two main categories, exotoxins and endotoxins.
Exo and endo. Let's start with exotoxin.
Okay, exotoxins are proteins. They're produced inside some bacteria, can be gram-positive or gram-negative, and then they are actively secreted or released outside the bacterial cell, often when it lyses.
So they're deliberately released weapons.
Pretty much. And they are often enzymes. This is key because enzymes can act over and over again, catalyzing a reaction multiple times. That means even tiny amounts of an exotoxin can be incredibly potent and damaging.
Small amount, big effect.
Exactly. They're also highly specific. They target particular parts of host cells or inhibit very specific metabolic functions. We mentioned botulinum toxin earlier. Just 1 milligram can kill 1 million guinea pigs. That's specificity and potency.
Wow.
And the specific signs and symptoms of diseases like diphtheria, tetanus, botulism, scarlet fever, those are primarily caused by these exotoxins.
Is there any good news about exotoxins?
Yes.
Yeah.
Because they are proteins, our bodies can produce specific antibodies against them, called antitoxins. These antibodies can neutralize the toxin. And even better, we can inactivate exotoxins, treat them with heat or chemicals to create toxoids.
Toxoids.
Yeah, inactivated toxins that are no longer poisonous but still stimulate an immune response. Toxoids are the basis for vaccines against diseases like diphtheria and tetanus. They give us immunity.
That's fantastic.
Yeah.
Using the weapon against itself to build defense. So what are the different kinds or categories of these potent exotoxins?
They're broadly categorized into three main types based on their structure and function. First, you have AB toxins. Yeah, they consist of two parts, an active A component, which is the enzyme part that causes the damage, and a binding B component, which attaches the whole toxin to a specific receptor on the host cell surface and helps get the A part inside.
So B is the delivery system, A is the payload.
Perfect way to put it. Diphtheria toxin is a classic AB toxin. It inhibits protein synthesis in our cells, particularly damaging nerve, heart, and kidney cells. Some nasty gram-negative bacteria even make AB toxins called genotoxins, which actually cause breaks in our DNA.
DNA damage. That sounds bad.
It is. Can lead to mutations, cell cycle disruption, potentially even cancer down the line. Second type, membrane-disrupting toxins.
What do they do?
These guys cause lysis, they rupture host cells, they do it either by forming protein channels or pores right through the plasma membrane, making it leaky.
Like punching holes?
Exactly. Or by disrupting the phospholipid portion of the membrane itself, destabilizing it. Examples include leukocidins, which specifically target and kill leukocytes or white blood cells, including phagocytes.
Weakening the immune defense directly.
Right. And hemolycins, which destroy erythrocytes, are red blood cells. Streptolacins, made by Streptococci, are famous hemolycins that can lyse both red and white blood cells. Some intracellular pathogens, like Listeria, cleverly use these toxins to break out of the vacuoles they get trapped in once inside phagocytes.
Escape artists. Okay, third type.
Third type are superantigens. These are quite different.
Super antigens. What makes them super?
They provoke a very intense but completely nonspecific immune response. Normally, only a tiny fraction of our T cells, a type of immune cell, would respond to a specific conventional antigen. But superantigens bypass the normal process. They essentially force a connection between immune cells that shouldn't be there, stimulating a huge number of T cells all at once.
Like pressing the alarm button for the whole building instead of just one floor.
Excellent analogy. This massive T cell activation causes them to release enormous amounts of cytokines.
Cytokines, those are the immune system's chemical messengers.
Yes, and normally they're tightly regulated. But a flood of cytokines, a cytokine storm, causes systemic effects. High fever, nausea, vomiting, diarrhea, sometimes shock, organ failure, and even death.
Where do we see this happen?
Staphylococcal toxins that cause food poisoning and toxic shock syndrome are classic examples of superantigens.
Scary stuff. Okay, that covers exotoxins, secreted proteins, specific targets, often enzymes, can make toxoids.
Yeah.
Now, What about endotoxins? How are they different?
Endotoxins are fundamentally different and in some ways maybe more insidious. First off, they are not secreted proteins.
Okay, non-secreted. What are they then?
They are the lipid portion, specifically lipid A of lipopolysaccharides, LPS.
LPS. That rings a bell. Outer membrane.
Exactly. LPS is a major component of the outer membrane of gram-negative bacteria only. Gram-positives don't have it.
So only gram-negative bacteria produce endotoxin.
Correct. And crucially, the endotoxin, the lipid A, isn't actively secreted as part of the bacterial structure. It's primarily liberated when the bacteria die and their cell walls lyse or break apart.
Released upon death. That seems important.
It's critical. It means, for example, that when you treat a severe gram-negative infection with antibiotics that kill the bacteria, you can sometimes get an initial worsening of symptoms.
Because killing the bacteria releases a flood of endotoxin.
Precisely. A temporary endotoxin surge.
What kind of effects do endotoxins cause? Are they specific like exotoxins?
No, they're much more general. All endotoxins, regardless of which gram-negative species they come from, tend to produce similar systemic effects. Things like chills, fever, what we call the pyrogenic response weakness, generalized aches. In severe cases, it can lead to shock, potentially disseminated intravascular coagulation, DIC, and even miscarriage.
How do they cause fever?
They stimulate our macrophages, immune cells, to release those cytokines again, particularly interleukin-1, IL-1, and tumor necrosis factor alpha, TNF-alpha.
The same messengers involved in superantigen response.
Yes, but triggered differently here. These cytokines travel through the blood to the hypothalamus in the brain, our body's thermostat. There, they trigger the production of prostaglandins, which effectively reset the thermostat to a higher temperature. Hence, fever.
Okay. And the clotting issue? DIC.
Endotoxins also activate blood clotting proteins inappropriately. This can lead to the formation of numerous tiny blood clots throughout the small blood vessels. This uses up clotting factors and can paradoxically lead to bleeding, but it also obstructs blood flow, causing tissue damage or death due to lack of oxygen. That's DIC.
That sounds incredibly dangerous.
It is, and perhaps the most severe consequence is septic shock.
Shock caused by infection.
Specifically, often by endotoxin from gram-negative bacteria. TNF, released in response to endotoxin, increases the permeability of our capillaries, our smallest blood vessels.
Makes them leaky.
Exactly. Fluid leaks out of the bloodstream into tissues, causing a massive drop in blood volume and therefore blood pressure. This severe drop in blood pressure impairs blood flow to vital organs, leading to organ failure. It's a life-threatening emergency.
And can we make antitoxins against endotoxins like we can for exotoxins?
Unfortunately, no. Endotoxins are lipids, not proteins, and they don't effectively stimulate the production of neutralizing antitoxins. This makes developing lasting immunity or effective antibody-based treatments very difficult.
So they're harder to neutralize. How do we even detect them then? Especially if they can contaminate things that are supposed to be sterile, like IV fluids or drugs.
That's a great question and leads to a fascinating bit of biology. We use the Limulus amoebocyte lysate assay, or LAL assay.
Limulus, horseshoe crab.
Exactly. The assay uses lysate, basically, broken open cells from the amoebocytes, which are the blood cells of the Atlantic horseshoe crab, Limulus polyphemus.
What do crab blood cells have to do with endotoxin?
It turns out that these amoebocytes contain proteins that clot very rapidly and intensely in the presence of even tiny amounts of endotoxin. It's part of the crab's primitive immune system.
So if the lysate clots, there's endotoxin present.
Precisely. A positive LAL test indicates the presence of endotoxin. It's incredibly sensitive and is used routinely to test sterile injectable drugs, medical devices, IV solutions, anything that bypasses the body's natural barriers to ensure they aren't contaminated with endotoxin from dead gram-negative bacteria.
Even if the solution was sterilized?
Yes, because sterilization might kill the bacteria but won't necessarily destroy the heat-stable endotoxin they release. This assay was actually key in solving a clinical mystery where patients developed severe eye inflammation after cataract surgery.
What happened?
The enzymatic solution used during the surgery, although sterilized, was found to be contaminated with endotoxins, the source. Dead Burkholderia bacteria that had grown in the purified water pipes used to make the solution.
Yeah, a stark reminder that sterile doesn't always mean endotoxin-free.
Absolutely. It highlights why rigorous cleaning, purification, and testing protocols are just paramount in healthcare and pharmaceutical manufacturing.
Okay. So much of this ability to cause disease, this pathogenicity, seems built into the microbe's biology capsules, enzymes, toxins. But you also hinted that genetics plays a role. How do microbes actually acquire these virulence factors? How are they shared or picked up?
Right. This is where microbial evolution gets really dynamic and, frankly, a bit alarming. Two key mechanisms are plasmids and lysogeny.
Plasmids, those are the extra little circles of DNA, right?
Exactly. Small circular DNA molecules separate from the main bacterial chromosome. Think of them like tiny portable genetic accessory packs or USB drives.
Carrying extra software.
Good analogy. They often carry genes for things like antibiotic resistance. We call those R factors. But crucially, they can also carry genes encoding major virulence factors.
Like what kind of factors?
Genes for tetanus neurotoxins, certain enterotoxins that cause diarrhea, some adhesins that help them stick, coagulase that forms clots. Lots of key weapons can be plasmid-borne.
So a harmless bacterium could potentially pick up a plasmid and become dangerous.
It absolutely can. That's horizontal gene transfer in action. Now, the second mechanism is lysogeny.
Isogeny.
Yeah.
That involves viruses, right?
Yes, specifically bacteriophages or phages, which are viruses that infect bacteria.
Viruses infecting bacteria.
Okay. Sometimes when a phage infects a bacterium, instead of immediately replicating and killing the host, it integrates its own DNA directly into the bacterial chromosome. This integrated phage DNA is called a prophage.
So the virus's genes become part of the bacterium's genes.
Essentially, yes, and this can lead to lysogenic conversion. The bacterial host cell can acquire entirely new properties, new traits that are encoded by the genes within that prophage DNA.
And some of those new traits can be virulence factors.
Many important ones are, actually. The genes for diphtheria toxin, botulinum neurotoxin, cholera toxin, the pyrogenic toxins causing scarlet fever, even the dangerous Shiga toxin found in E. coli O157. All of these are carried on prophages.
So the bacteria are only dangerous because they got infected by a specific virus.
In these cases, yes. The phage provides the blueprint for the toxin. It's a remarkable example of how genetic material can move between different biological entities.
It really drives home how fluid microbial genetics are. It's not just about inheriting traits, but acquiring them sideways.
Exactly. And sometimes these mobile genetic elements can jump between different bacterial species too, spreading virulence or resistance traits rapidly through microbial populations. Think about MRSA, methicillin-resistant Staph aureus. Some of its enhanced ability to colonize skin might come from genetic elements acquired from normally harmless skin bacteria like Staphylococcus epidermidis.
Wow. This constant evolution in gene swapping is what makes things like antibiotic resistance such a huge ongoing challenge.
It really is. They're constantly adapting, constantly finding new ways to survive and thrive, often at our expense.
Okay, bacteria are clearly ingenious and often quite dangerous. But they're not the only players in this game, are they? Viruses, fungi, protozoa, helminths, even some algae. They all have their own ways of causing trouble. Let's touch on viruses first. How do these, well, non-living entities cause disease?
Right. Viruses are fascinating because they're obligate intracellular parasites. They have to get inside our cells to replicate. This gives them a major advantage. They can hide from many components of our immune system simply by being inside our cells.
Out of sight, out of mind for the immune system.
To some extent, yes.
Yeah.
They get in by having specific attachment sites on their surface that bind very precisely to receptors on our target host cells. Again, that lock and key mechanism.
Do they have clever ways to bind?
Some do. The rabies virus, for example, has attachment sites that apparently mimic acetylcholine, a neurotransmitter, helping it bind to nerve cells. HIV is particularly cunning. It hides its main attachment sites deep within folds or valleys on its surface, making it physically difficult for our large antibody molecules to reach and bind them. Plus, of course, HIV directly attacks and destroys critical immune cells, the T helper cells.
A direct assault on the command center. Once they are inside, how do they cause damage?
Once inside, viruses take over the host cell's machinery for their own replication. This process itself often damages or kills the cell. We see visible changes in infected cells called cytopathic effects or CPEs. These are really important for diagnosing viral infections in the lab.
What kind of changes are CPEs?
They can range quite widely. Stopping normal cell division, causing the cells to lyse or burst open, releasing new viruses, forming characteristic clumps or structures inside the cell called inclusion bodies, like Nigri bodies, seen in rabies-infected brain cells. Sometimes they cause infected cells to fuse together, forming large multinucleate giant cells called syncytia. We see that with viruses like measles and mumps.
So the virus physically changes the cell's appearance and function.
Yes. Viruses can also shut down the host cell's own synthesis, induce new antigens on the cell surface, which can mark the cell for destruction by our immune system, sometimes cause chromosomal damage, and in some cases transform host cells into cancerous cells by disrupting normal growth regulation.
And do they have ways to fight back against our defenses, like interferon?
Oh, yes. While our cells produce interferons as an early warning signal to fight viral infections, almost all viruses have evolved mechanisms to block interferon synthesis or its action. They try to disarm that alarm system right away.
Always one step ahead, it seems. Okay, what about the others? Fungi, protozoa, helminths, algae. Do they use similar tricks, or are their methods completely different?
They each have their own distinct pathogenic properties, often reflecting their more complex biology compared to bacteria or viruses. Let's take fungi.
Okay, fungi, like molds and yeasts.
Right. Many fungi produce metabolic byproducts that happen to be toxic to us. For instance, molds like Fusarium or Stachybotrys, the black mold, can produce trichothecenes. These toxins inhibit protein synthesis and can cause severe gastrointestinal issues if inhaled or ingested.
Toxic mold.
Yes. Or Claviceps purpurea, a fungus that grows on rye and other grains. It produces ergot alkaloids. Ergot poisoning historically caused hallucinations, convulsions, and gangrene of the limbs.
Wow.
And Aspergillus flavus, a mold that grows on peanuts and corn, produces aflatoxin, which is a potent carcinogen strongly linked to liver cancer.
So toxic byproducts are a big weapon for fungi. Do they have other virulence factors?
Some do. Certain fungi, like Candida albicans, which causes thrush, produce proteases, enzymes that help them attach to and invade tissues. Some, like Cryptococcus neoformans, a cause of meningitis, have thick polysaccharide capsules that resist phagocytosis, just like some bacteria. And chronic fungal infections can also trigger allergic responses in the host.
Okay, what about protozoa? Single-celled parasites like Giardia or the malaria parasite.
Their presence in waste products often directly cause symptoms. Plasmodium, the malaria parasite, is famous for invading our red blood cells, multiplying inside, and then rupturing them, leading to those characteristic cycles of fever and chills.
Destructive cycle.
Very. Giardia intestinalis, which causes diarrheal disease, attaches firmly to the cells lining our intestines using a specialized sucking disc, and it appears to digest the cells directly, causing damage and malabsorption.
Physically damaging the gut lining.
Yes. And like some bacteria and viruses we discussed, some protozoa are masters of antigenic variation. Giardia can do it, and Trypanosoma, the sleeping sickness parasite, is the ultimate example, constantly changing its surface coat to evade our immune system for years, even decades.
Persistent infections. Now, helminths, these are the parasitic worms, right?
Yeah.
Much larger.
Much larger, yes. Because of their size, they often cause disease simply by using host tissues for their own growth, forming large parasitic masses, or physically blocking anatomical pathways.
Like a physical obstruction.
Exactly. Wuchereria bancrofti, the worm that causes elephantiasis, lives in and blocks lymphatic vessels. This blockage prevents proper fluid drainage, leading to that grotesque, massive swelling of limbs or other body parts. And like other parasites, their metabolic waste products can also contribute significantly to the host's symptoms and inflammation.
And lastly, algae. I don't usually think of algae as pathogens.
Most aren't, but a few species produce incredibly potent neurotoxins that can harm humans, usually indirectly.
How so?
Dinoflagellates, tiny marine algae, sometimes form massive blooms like red tides. Some species, like Alexandrium, produce saxitoxin. Shellfish, like clams and mussels, filter feed on these algae and concentrate the toxin in their tissues. If humans then eat these contaminated shellfish...
Paralytic shellfish poisoning.
Exactly. Saxitoxin blocks nerve transmission, causing paralysis, potentially respiratory failure, and death. Another group, diatoms, like Pseudonychia, can produce domoic acid, again, concentrated by shellfish. Eating contaminated shellfish leads to amnesic shellfish poisoning.
Amnesic.
Yes, it causes neurological damage, including permanent loss of short-term memory, seizures, coma, and can even be fatal.
Yeah.
So while not direct infections, these algal toxins are a serious pathogenic threat mediated through the food chain.
A whole different kind of microbial danger.
Yeah.
Okay, so we've journeyed through how microbes get in, how they stick, how they evade our defenses, how they cause direct damage or use toxins. But to really understand their life cycle and how diseases actually spread, we need to know how they get out. What are the common portals of exit?
Right, the exit strategy. Just like they have preferred ways in, microbes also typically leave the body via specific portals of exit. These exits are usually related to the part of the body that was infected.
Makes sense. Where they lived is where they leave.
Generally, yes. They leave in secretions, excretions, discharges, or sometimes in shed tissue. And often the portal of exit mirrors the portal of entry.
So what are the most common exits?
By far the most common are the respiratory and gastrointestinal tracts.
Coughing, sneezing, feces.
Exactly. Respiratory pathogens, like those causing tuberculosis, influenza, measles, whooping cough, they're discharged in droplets from coughing or sneezing. Gastrointestinal pathogens like Salmonella, Shigella, Vibrio cholerae, amoebas, they exit in feces or sometimes saliva.
Okay, what else?
The genitourinary tract is another important portal of exit, mainly for sexually transmitted infections found in semen or vaginal secretions, but also some other bacteria can be shed in urine. Skin infections can spread through pus or drainage from lesions, or simply by direct contact with infected skin or shed skin cells.
And blood.
Yes, blood can be a portal of exit, but usually requires assistance. Biting insects, like mosquitoes from malaria or yellow fever, act as vectors, taking infected blood from one host and transmitting it to another. Or contaminated needles and syringes can transfer bloodborne pathogens like HIV or hepatitis B and C.
So the exit route is crucial for transmission to the next host.
Absolutely. Understanding portals of exit is fundamental to preventing the spread of infectious diseases, things like covering coughs, hand washing after using the restroom, safe sex practices, controlling insect vectors, ensuring sterile medical procedures. It all ties back to blocking these exit and subsequent entry routes.
What an incredible and slightly terrifying journey through the unbelievably sophisticated world of microbial pathogenicity.
It is quite something.
From the really cunning ways they, sneak in and manage to stick to our tissues, to the ingenious strategies they deploy to dodge our complex defenses, and those potent toxins they use to wreak havoc.
Yeah.
It's just clear that the microscopic world is full of strategic masterminds, constantly evolving right alongside us.
Indeed. And understanding these intricate mechanisms, all these strategies and counter-strategies, it's absolutely fundamental to everything we do in medicine and public health. How we develop effective treatments, how we design vaccines, how we prevent the spread of disease. It all comes back to understanding this microscopic battle.
Right.
And this whole intricate co-evolutionary dance between us, the hosts, and the microbes. It really raises an important question for all of us to think about, doesn't it? What does this continuous microscopic arms race tell us about the future of medicine, especially as microbes continue to evolve, continue to adapt, constantly presenting us with new challenges, but maybe also new opportunities for discovery?
That's a great point. Something for all of you listening to definitely mull over. We really hope you've enjoyed this deep dive into the microbial mechanisms of pathogenicity.
Yeah, hope it was insightful.
Until next time, keep exploring, keep learning, and keep being well informed.